On behalf of the Brain Tumour Charity, I'd just like to thank everybody for coming this evening, and it's a good turnout. I'd also like to thank Queen Mary's for hosting the event, and to thank Denise Shear, Surinder Pal and Professor Silvia Marino, who's not here tonight, who helped organise it in conjunction with David Wixman. We're very pleased to have Dr. Colin Watts here tonight. He's got time out from his busy schedule to be here. Colin is a neurosurgeon at Addenbrooke's Hospital, part of Cambridge University. He also has an academic interest in brain tumours, and over the last number of years he's really made Cambridge one of the leading research institutions in the UK. He's also the chairman of the Brain Tumour Clinical Trials group, and in conjunction with us as a charity, we're desperately trying to get more patients through to clinical trials, adult patients through into clinical trials in the UK. As you might be aware, less than 2% before Colin became chairman go through onto a clinical trial in the UK, whereas with breast cancer, 38% of patients go through to clinical trial. And in conjunction with Colin, we really are making progress on that front. And he is leading from the front. He's the chief investigator of the GALA5 clinical trial project for glioblastoma that's funded by us and Cancer Research UK, and his team at Cambridge have also recently got funding for another 2 clinical trials. So things are really progressing, and so I'd like to hand over to Colin, and he's going to give us an in-depth information on brain tumours and his neurological experience as well. Thank you.
Thanks a lot, Neil. Thank you very much. So my brief today is to talk a little bit about impact, a little bit about management, and a little bit about what the future holds. So what I want to do is I want to begin by discussing some of the issues that have arisen over the years. Our fundamental philosophy is that good research is predicated on having clinical excellence. You can't have good research without good clinical care. And one of the issues that was to try and establish the kind of clinical infrastructure. And we've succeeded to a level that is probably unique in the UK at the moment, but we are now in a process whereby we are trying to apply the lessons we've learnt in Cambridge to other centres, particularly from a neurosurgical perspective. So one of the things that we've been doing is to ensure that we've established a dedicated process of referral to a specialist multidisciplinary team. The emphasis is on team management. It's not just me, it's not just an oncologist. It's all of us and the nurses and the support workers all working together. Then we needed a first point of contact, and that's what we call the glioma clinic. In other words, every patient with a brain tumour in the Anglian Cancer Network comes through our clinic. That's one clinic. It's manned by 3 consultant neurosurgeons. It runs 52 weeks a year. If one consultant goes on holiday or comes to do a talk like this, the rest of them carry on covering the clinic. And the other thing that's extremely important is ensuring that we've got effective, empathic, efficient patient care. And that's extremely important. So it demands a multidisciplinary team-based approach, and our model is based on the improving outcomes guidance. What does it involve? Well, it means we try to understand the patient journey. We try to understand what happens to patients from the point at which they get picked up and put into the system. And we wanted to develop a dedicated pathway that they could follow, so every patient is managed in the same way. We wanted to establish a neurosurgical oncology clinic, and we've succeeded in doing that. It's the only one of its kind in the UK, but others are beginning to look quite closely at how we do things. But most importantly is that we managed to build a clinical team. And this is the pathway such that the point of initial referral then goes into a standardised pathway of care that is safe, so that when a patient with a lump in their head is referred, they are managed in the same way. If we think it's a tumour of some description, we need to make sure it's not something like an abscess, which would be a surgical emergency. So they're screened, which is what the consultant review of the scans is all about, and then they go through this pathway. And so that we understand now, from point of referral to the oncology or palliative care, we understand this patient pathway. And one of the new initiatives that we're just trying to develop is to understand what happens out here. In other words, what is it what patterns of symptoms can we identify that will help us detect and pick up the disease earlier? And so we've got an application for some funding in to look at that in the context of other rare cancers. Now, as some of you are well aware, because I keep banging on about this, brain cancer has a disproportionate burden of disease. When I ask friends and people at meetings and trainees and students, can they name someone with breast cancer? Usually they come up with something fairly quickly, usually Kylie Minogue actually. If I ask them, can they name anyone with brain cancer, quite often they're stumped. And yet there are lots of famous people. There are people like Sevi Ballesteros, there are people like Ted Kennedy, and for those of you who are a little bit older than me, Emlyn Hughes. There are lots of people, but the burden's poorly understood. Brain cancer accounts for 2% of cancers as we know, but 7% of cancer deaths. Astrocytic tumours are the third leading cause of cancer-related death in middle-aged men. And they're the fourth leading cause of cancer death among women aged 15 to 34. And this is because the other cancers are receiving treatment. They're getting better treatment. And pilocytic astrocytomas and medulloblastomas are the commonest solid tumour in children. So back to that pathway and the implementation of the IOG guidance. And basically, prior to implementation of that guidance, practice was basically consultant-centred. It was Mr. So-and-so's clinic. When you go for a consultation in the hospital, you go to Mr. So-and-so's clinic. They're patient-orientated. I'm not suggesting for 1 minute that they are not doing their very best for patients. I'm sure they are. But it's research ambivalent. Because if you have 3 different consultants all dealing with brain tumours, all on different days, and you happen to go to Mr. A's clinic, he'll only see a third of all the referrals, and therefore the chances of picking patients up, getting them into trials, is much more difficult. Patients were seen by the on-call team. You're referred in, and whichever team happens to be on call picks it up. Clinical management was variable, and surgical specialisation just didn't exist. And there was a lot of therapeutic nihilism. Oh, there's no point being too aggressive because patients are going to die. And as I'll show you later on, that's just not true. And as I've already mentioned, few patients could be entered into clinical trials. So we've shifted things. Now it's patient-centred. You don't come to Mr. So-and-so's clinic, you come to the glioma clinic. It's consultant-led. There are 3 consultants who support that clinic. We're not all 3 there all the time, but that clinic runs with a consultant 52 weeks a year. And it's research-orientated. We try and get every patient into some sort of clinical trial or research study. And if you include tissue banking, our research, the number of patients going into research now is of the order of 80% of patients going through clinic. For patients, it means the consultants know what they're talking about, and they're genuinely interested. And we take time to discuss the disease and allow patients to go home, think about what's happening, think about what treatment options there are. So the process of consent is enhanced. Consent is a process, it's not an event. You don't go to a clinic, give your consent and then move on. You need to think about it. You need to try and understand what's happening to you. It's very scary if you're a patient. They meet a dedicated specialist nurse who can ask all of the things, silly questions. We have a philosophy in clinic which is that there's no such thing as a silly question. There are plenty of stupid answers, but there's no such thing as a stupid question. We try and optimise clinical management. We don't get it right all of the time, but we try and get it right as much of the time as we can, and this for us is a key fundamental philosophy. So, we published a paper 2 years ago now where we looked at 2 periods, one before we got the clinic going and one after, and what we found is that our survival statistics were as good as or better than. This is the Stuck trial for patients with glioblastoma. This is if you get radiotherapy. This is if you get radiotherapy and chemotherapy. The black line here is Addenbrook's radiotherapy, and the green line is our radiotherapy and temozolomide. And this is our data compared to the Stuck data. Now, I'm not saying we're better. Our cohort is smaller, so the statistics don't weigh up. But what is true is that our 5-year survival is now 16% for patients with glioblastoma. So we've established a high-quality service. We've improved the procedure and process of consent. We've got procedure-specific consent forms. Management plans can be established early, patients can get appropriate information and counselling, and you leave the clinic with a date for surgery. So you know the plan, and you've got time to come to terms with it. Governance and audit, it's an important thing in the NHS. North Staffs illustrates what happens if this is not rigorous. And we facilitate communication between the subspecialties. We can introduce different elements of service development, which I'll come back to. And so, as I said, it's a team effort. So pre-operative planning has gone up from 2/3 to it's over 90% now. Median length of stay, the time patients spend in hospital has been halved. Patients don't like being in hospital. Hospitals are dangerous places. They're full of doctors for a start. Histopathology, reporting time halved. Every patient is given a diagnosis in a specialist clinic now, with the opportunity to spend as much time as they like with a specialist nurse and a doctor if that's necessary. The time from surgery to diagnosis has gone down significantly, it's nearly halved, and this has been the biggest jump. Post-operative MRIs have gone from 17 to over 90%, and I will come back to that. And the feedback's been largely positive. We don't get it right all of the time, and there's always room for improvement. Going forward, what are we interested in? We're interested in molecular diagnostics. I'll come back to that, but basically it's trying to understand in more detail the biology of what disease you have and tailoring the treatment to that disease. We want to improve the efficiency of service delivery. Telephone clinics are now established. Most post-op MRs are actually we do get within 48 hours or 72 hours at the absolute latest. And in some cases, patients come in on the day of surgery, have their surgery and go home the next day. Additional imaging, all this fancy stuff that I don't really understand a lot about, but basically MRI tracks, looking at the fibre pathways, looking at what's happening, that rather than anatomical, structural imaging, imaging function, imaging the pathophysiology. Evaluating quality of life. There's no point us doing masses and masses of high-tech stuff if your quality of life is worse. There's just no point doing that. And another area that is a growth industry in more ways than one is brain metastases, no pun intended there. So these are the areas that we're targeting, and innovation particularly is something that we're interested in developing. As Neil said in his introduction, there's more to it than simply doing good clinical service. There's a lot of politics going on, which he's only too well aware of, but it's important about raising awareness of brain cancer. That's one of your fundamental missions, the Brain Tumour Charity's fundamental missions. We have representation on most national bodies now, and we interact with policymakers. And our most recent one has been to work with CRUK and the National Cancer Intelligence Network to improve the statistics, because one of the problems that is, as I'll come back to, is that grade 2 tumours in adults are classified as a low-grade and benign disease by the Office of National Statistics. The median survival, as I'll come on to, for a patient with a low-grade tumour is about 5 years. And if that was a breast cancer, that would be classed as an aggressive cancer. So we've been working with CRUK to get accurate statistics with rigorous classification of malignant phenotypes. And that website will go live on the 28th. You'll be able to access it on Cancer Researchers' web pages. So we're beginning to try and define what we're tackling. Brain cancer is expensive. It costs Europe about 5.2 billion euros based on 2010 data, of which we're about 800 million. That's a lot of money. The average cost of a high-grade cancer is about 29,000, and a low-grade and a meningioma, similar. It's expensive. So are patients getting value for money? Good question. This paper was published in 2009, uses Eurocare data. Now, Eurocare is a big European-wide registry. And what they did is they looked at patients diagnosed between 1995 and 1999. That's a 5-year cohort. And they then followed it forward to 2003, so that over that period they looked at the deaths. And then they adjusted those deaths for the background deaths in the population generally. And they then came up with a statistic of avoidable deaths. And they did that looking at two elements. One was the mean European survival. So what would happen if we got our treatment as good as the average in Europe? What would happen if we got our treatment as good as the best in Europe? So if we got it as good as the average, we would save 59 patients every year, because there are 59 people who die every year who would not die if we were as good as the average in Europe. If we were as good as the best in Europe, 225 patients would not die every year. So, are we getting value for money for all this money we're pouring in? I don't think so. I don't think you'd agree. I think you'd agree with me that that's not good value for money at all. What about prognosis? One of the things that has been annoying me for some time is that a lot of the statistics that are quoted are based on the results for patients who get optimal treatment. And that's only a fraction of the whole patient population. And the patient population statistics are actually rather different, and there's very little data out there. There's one Swedish study which was published in 2005, not Swedish, Swiss, and they looked at the survival of everybody who was diagnosed with a grade 4 tumour. And that survival was 0.4 years. That works out at 4.6 months. When I ask my trainees and students what is the median survival for a grade 4 glioblastoma, they quote me 14 months. Because 14 months is the median survival for the patients who got optimal resection, optimal radiotherapy, optimal chemotherapy. And for a lot of patients, that just isn't possible. What about an anaplastic astrocytoma, a grade 3? 1.6 years. And this is my favourite, what the Office of National Statistics regard as a benign tumour. Median survival 5.6 years. Varies enormously, we'll come back to that, but 5.6, if that was breast cancer, there'd be a hoo-ha. That would be one of the most aggressive types of breast cancer you could get. Grade 1s are the only good news, really. Oligodendrogliomas, grade 3, 3.5 years. It makes a difference knowing what they are and being able to distinguish them. And if you've got a grade 2 oligodendroglioma, your typical survival is 11.6 years. And that's what it looks like if you graph it. So unless you've got a pilocytic astrocytoma, you know, the news isn't good. And yet these kind of figures are poorly understood, poorly recognised. So, a bit of biology. One of the things that Brain Tumour Charity have been keen advocates of is, along with a lot of other people, is patients being allowed access to tests, to biomarker tests. And this is why. What's a biomarker first? Let's define what we're talking about. Biomarker is anything that can be used as an indicator of a particular disease state. In neurosurgery, a biomarker is a measurable characteristic reflecting the severity or presence of a disease. And that applies to any branch of medicine. And they can be diagnostic, prognostic, or predictive. Diagnosis means establishing what you've got. Prognosis means establishing what's going to happen to you, irrespective of what treatment you get. And predictive is about understanding what happens if we treat you with a drug. So prognostic biomarkers are measurable characteristics signaling an effect on outcome independent of treatment. And predictive biomarkers indicate benefit from a particular treatment or intervention. Okay? So that's what we're dealing with. And these are the three that you may have heard about: IDH1, MGMT, and 1p19q. They're the ones that are clinically relevant, the ones that people struggle to get. And this is the relevance of them for grade 2, 3, and 4 tumours. So your IDH1 status has prognostic value in grades 2 and 3. It's rare in grade 4. MGMT isn't really, it's rare in grade 2s, and we don't really understand its function. But in grade 3, patients who have a methylated MGMT have a better prognosis if they're going to get chemo-RT. And for glioblastoma, where most of the data was generated, then it's predictive of benefit from chemo-RT. So if we know what this is, we can understand better how you're going to respond to the drug. And 1p19q co-deletion is good, it's prognostically favourable in grades 2 and 3 and is rare in grade 4. So that's just a snapshot, don't get too hung up about it, but that helps us begin to try and understand some graphs. This is IDH1. If you are mutant is good, okay? Which is unusual, it's usually the other way around, it's counterintuitive, but if you're mutated, that's a good thing. So this is the survival curve if you've got an IDH1 mutated tumour, and this is the survival curve if you're wild type. Wild type means it's normal. It's like it is in most of the population. And so this is comparing grade 3 and grade 4 tumours, whether they're IDH1 wild type or mutant. And the interesting thing is this, if you look carefully at this graph, you can see that a glioblastoma, a grade 4, that is mutant, has a better survival curve than a grade 3 that is IDH wild type. So if you're told you've got a grade 3, but you're wild type, we know that your prognosis is no better than a glioblastoma. What about age? Age we know is an important factor, and if you're less than 60, look at the survival curves. If you're an astrocytoma grade 3 and you've got an IDH1 mutation, your survival is good. If you've got a glioblastoma and you're wild type, which is what most glioblastomas are, it's bad. And if you're a glioblastoma at grade 4 or a grade 3, there's no different. If you happen to be one of that small number of glioblastoma with an IDH1 mutation, i.e. secondary glioblastomas. If you're over 60, we don't have as much data, but you don't have to be a brain surgeon to work out that the survival curves for grade 3 and 4 are the same. Okay? So at the moment what we do is we manage patients based on whether they have an anaplastic astrocytoma, that's a grade 3, or a glioblastoma, that's a grade 4. And that is diagnosed by very clever pathologists looking down a microscope. It's called a morphological diagnosis. But if we can shift to treating patients not morphologically, but based on some of the biomarkers, some of the biology of the disease, then we'll start to change what happens. What about MGMT? Well, I've already told you that if it's methylated, that's good. It will respond well to treatment with alkylating temozolomide, basically. And there's not much difference there, just illustrating that in terms of response to temozolomide, methylation is a good idea. 1p19q is an interesting one because that tends to occur in oligodendroglial tumours, the ones with a better prognosis. And initially what happened is these are patients who have a deletion, and these are patients who do not have a deletion. And this is, and look at the scale, this is years. But what we found, and it's been published earlier this year, is that if you are deleted and you have radiotherapy and a chemotherapy, your survival suddenly, we realise, is suddenly much better than if you just get radiotherapy alone. But that survival benefit doesn't really manifest itself until well beyond 7 years. And if you look at the median, the halfway mark, and you look across, the median survival for patients with a 1p19q deleted oligo who get radiotherapy is about 10 years, ballpark. But if you look at the curve for those getting PCV, it doesn't reach the median. And we're out now to 14 years. So we really are beginning to understand that biology can make a difference in terms of what the treatment effect is. If you're 1p19q not deleted, it doesn't make much difference. There's a little bit of benefit for chemotherapy. It's not the same. How is surgery helping? Well, there's been this recent technology which we've taken up quite a lot and are pushing, in which we cause the tumour to glow pink. What happens is a patient has a pink drink before surgery. It's not pink, it's clear. It tastes a little bit bitter. But what it does is it causes the tumour to glow pink under specific operating conditions. And what that does is it allows us to begin to distinguish what we thought was normal brain we actually realise is actually riddled with disease. And I'll come on to this again in a minute. So what we've done is we've found that if you use fluorescence, you get a better resection. If you get a better resection, you have less contrast-enhancing residual disease. You leave less disease behind. And what this data shows is that if you use 5-ALA, the number of patients with residual contrast-enhancing disease falls from about 80% down to around about 50%. And the patients with no contrast-enhancing disease goes up. And I'll come back to that in a minute. I think, yeah, this is why it's important. If there is no residual tumour, and it's binary, it's either no residual or residual, then there is an impact on survival. That's prospective trial data on a large series. That's what's happening at Addenbrooke's. These are patients with no residual disease. These are patients with residual disease, glioblastoma. So it makes a benefit. And part of the reason, I have to turn a film down here. Part of the reason is it is beginning to help us understand how the tumours spread. So what I want you to pick up is that there's white matter here that connects one gyrus to another. And we know that these white matter tracks, these fibre tracks here, spread throughout the brain. And they act like little highways for the tumour cells to migrate along. And we know from some of the fancy imaging that I mentioned that we're getting a much more detailed understanding of how these pathways are organised, because it's these pathways that the tumour cells migrate along. And this is a surgical specimen. This is the grey matter on top. This is the surface of the brain, which would look essentially normal. And this is the white matter, which is riddled with disease, migrating from one gyrus across to the next. This is a patient with a tumour on the left side. MRI scans are always swapped round. This is the left side, and they had problems with speech. So we elected to do an awake procedure, and we elected to use the 5-ALA fluorescence. And this is a surgical shot after we'd just started. This is a part of the brain. It's one of the folds. It's called a gyrus. This is another one. So you've got gyruses or gyri, and then in between them you've got the dips, which are called sulci, and that's a sulcus. And what we've done is we've cut open the membranes that hold it all together. And we're beginning to see that there's something going on down here. I don't know if you can see that, but that's a little bit of fluorescent disease. And so what we do is we begin to extend the dissection, and we carry on the surgery, and we identify more of this disease, and it becomes clearer. Now what I want you to try and appreciate is that there is a bit of fluorescence here in this gyrus, which to all intents and purposes looks plumb normal. If we were doing this under a general anaesthetic, without fluorescence, we would leave that behind, because we know we're in eloquent brain. We know that the difference between there and there can make a difference between somebody being able to speak at the end of surgery or not. So we carry on, and we can see that the disease is actually spreading down here as we go deeper in. And yes, it's still there. There's still some in that other gyrus. So what we have to do is we begin to operate on the next valley, if you like. So we've gone over the hill into the next valley. And what we find as we operate is that the disease in what looks like normal brain, what we would never have touched, is actually riddled. So we can resect it. And we can extend our dissection around here. This is going round the corner. And again, this all looks plumb normal. Take my word for it. It's plumb normal, but it's not. So we can clearly see the distinction between invading infiltrative high-grade cancer and what we hope is normal or normal-ish brain. So at the end of the day, we achieve this, which is removal of this amorphous pink blob. What that allows us to do is it allows us to say, okay, we've taken all this out, we can stop now because it's gone. And we can actually begin to sort of interrogate. We can take samples, if it's safe to do so, of this non-fluorescent brain to see what happens, because one thing I want to make absolutely clear, just because we can achieve this kind of resection doesn't mean we're curing the patient. We know that there is residual disease there, but it's behaving differently to this stuff. I'll come back to that as well. So at the end of the day, what we achieve is this resection, and you can almost convince yourself that the outline of this resection cavity matches the outline of that contrast-enhanced disease. You can if you want to pat yourself on the back as a surgeon. But the post-operative imaging, it's gone. And that patient was awake throughout their surgery, and their speech was no worse at the end to what it was when we went in. This is our survival statistics, or we're beginning to look at a retrospective review of about 163 patients, which is one of the largest series in the world at the moment, where we're looking at patients who have residual contrast-enhancing disease and no residual contrast-enhancing disease. And as we look at these, and I can, your eyes might glaze over, but I want you to think back to a slide I showed you earlier. We can only tell whether we've got residual or no residual because we can get a post-operative MR. And a lot of centres still can't get a post-operative MR within 72 hours, which is what you need. We've gone from 17% post-operative MR to over 95% post-op MR. And because we've got that data, we can do this analysis. And what we're showing is that in patients who have no residual disease, who have a combination of chemoradiotherapy, their median survival is pushing towards 20 months. If they've got residual disease, it's still pretty good. It's nearer 15 months. So the surgery interacts with the treatment the oncologists give. It's part of the team approach. It's part of the holistic approach. So that's our clinical work. What about our research? Well, we're going to look at clinical research first. In other words, how's our pathway of care helping recruitment? And Neil mentioned recruitment is a key priority. So this is our pathway of care again. This time, we're looking at it from the perspective of clinical research and research trials. And what we can do is we can implement research studies at lots of different points along this pathway. And as I say, our latest one is out here. And also, we can get patients into clinical trials. That's the grand plan. And this is one of them, jointly funded by CRUK and the Brain Tumour Charity. The idea is to see if we use this fluorescence, if we use the pink drink, and we do good surgery, and we add in chemotherapy at the end of surgery, and then we hit hard with radiotherapy and chemotherapy, can we make a difference? That's essentially what we want to do. This is the recruitment for Gala 5. It's been up and running since the summer of 2011, and this is our recruitment. These are the total number of patients recruited, but some of them, for whatever reason, aren't eligible to be followed through, and this is the eligible number of patients, so we're spot on and hope to finish probably Q2 this year. Recruitment by centre. Again, going back to that pathway of care, the way we organise things, this is what we're doing. And that's what other people are doing. King's College, they have a similar system to us. Walton Centre also have a similar system. Other centres that don't have that kind of process of care in place struggle to recruit to UCLH. These guys are all new on the block. So Leeds are particularly enthusiastic. So those are our centres. This is our trial portfolio.
Does the national fall under UCLH?
Yeah. This is our trial portfolio. Some of these are open, some of these are pending, and Taverate, we're funding for scans was declined by the local research network, so we can't recruit for that. So we've got a busy portfolio, we've got a good pathway of care. What about the basic research? So at the end of the day, it's about developing a pipeline. And it's a two-way pipeline from the lab to the clinic and from the clinic back to the lab. So what we can do is we can go from bedside to bench. And what we're interested in is looking at perhaps thinking about how Charles Darwin might help us to understand why patients with glioblastoma suffer, why the treatment fails. I'll explain that in a minute. The fact that we've got good access to tissue means that we can start to look at how we can derive cells and develop model systems. And that's important. It's an important first step. And the reason is that we can get these tumour-initiating cells, stem cells maybe, maybe not, probably not. We know how to isolate them. We can get them now out of just about every glioblastoma that we operate on. And we are interested in trying to understand what the difference is between these cancer stem cells and these somatic stem cells. Somatic means normal. And the real difference, the one that matters, is these cause tumours. But there's an important distinction. And I don't want to labour the point, but essentially a cell of origin is different from a cancer stem cell. And it's the cancer stem cell that we want to try and target. All right? If we kill that, we try and kill off everything. And what that presupposes is it makes an assumption that the cancer, the way that the cells interact with each other, is organised in a hierarchy, like the army. So basically you've got the commander-in-chief who delegates. In other words, the commander-in-chief divides, regenerates themselves, and then produces another tumour cell. But it's these things that are important because they generate the tumour. They go on contributing to the growth and regenerating themselves. So we've spent a lot of time and a lot of effort looking at a variety of markers. The first one that came on the block was the idea of CD133. And what we've found is that both CD133 positive cells and CD133 negative cells can both generate themselves. So you can take a CD133 positive cell, generate positives and negatives, or a negative cell and generate positives and negatives. That means it's not a stem cell. The same applies to CD15, that's another marker, same thing. So then we started looking at it slightly differently. These are glial cancers. Maybe they come from glial progenitors. And the pathway, the biology, the basic biology of glial progenitors is very well understood, or at least it's more well understood than just about anything else. So we started looking at some of these markers that they use to identify glial progenitors, one of which was NG2. And we started looking at NG2. If you look at the difference between these graphs and that graph, it's completely different. Their behaviour is different. If you've got a positive cell, they'll generate positives and negatives. If you've got a negative cell, they'll generate positives and negatives, but they generate a lot as opposed to very little. If we look at patients, if we take patient samples and we look at the number of cells that are proliferating, that are dividing, because at the end of the day, a cancer, wherever it is a mass of cells that are dividing and are out of control. And we can measure that using this marker. And if we look at the number of cells that are dividing that express this marker, NG2, it's high. These cells are driving the tumour. But are they a stem cell? And the short answer is no, they're not. Because you can take a single negative cell or a single positive cell, you allow them to grow, and they will reform positives and negatives. So one negative cell will generate positives and negatives. One positive cell will generate positives and negatives. Not only will they generate positives and negatives, but they will generate positives and negatives in exactly the same ratio as the parent line they came from. So if the original line was 30% positive, 70% negative, then the cells that are regenerated from a single cell will be 30% positive, 70% negative. So there's no evidence for stem cell hierarchy in glioblastoma. So what's the alternative? Well, we've been looking at an alternative utilising this pink drink that we give patients. And we've been testing this idea, which is basically that the tumour that we take out is not the same as the tumour that we leave behind. To date, what's happened is people have taken these samples out, they've generated cell lines, They've used those cell lines to do experiments and test drugs and so on. And they've made the assumption that what they've taken out is the same as what's been left behind. So what we've been doing is we've been using our unique access to a lot of material to ask, well, is this margin, the M, the same as the T and the bits where it comes from? And what I want you to understand here is this is a log scale. So linear means they're growing very, very fast. And what we find is that these cells here from the margin aren't growing. They're dying off over about 2 or 3 months. They're dying off. Whereas these grow like weeds. So when we took these cells and we said, right, these are growing like weeds, these are not, they're dying. If we put those into a mouse, if we inject those cells into the brain, these will form tumours and these won't. And we were wrong. These form tumours, these form tumours. But the way the disease behaved was different. These are survival curves. These are tumours generated from this. Survival isn't good. Aggressive disease, rapidly growing. These are tumours derived from this and from the subependymal zone, and they are not as aggressive. They're still generating tumours. Remember, these are not stem cells. They're still generating tumours, but not as aggressively. So, I'll go back. So that holds. What we take out is not necessarily the same as what we leave behind. So what we've been doing then is we've been saying, right, okay, we can use this fluorescence to objectively sample. We can define a tumour because it's pink. And we can take spatially discrete samples. And that's how we do it during surgery in real time, sample from north, south, east and west. And the idea is that we have a separation of at least 10 millimetres between one place we take a sample and another. And then what we can do is we can look at all the, we can do some fancy studies looking at patterns of gene expression, copy number aberration and stuff. And what we did is we looked at these samples and we said, right, if all these samples come from, if the whole tumour comes from a common ancestor, then they should all have some common features that began with that common ancestor. So we took these multiple samples and we looked at the copy number aberrations and neutral somatic mutations. That's for your benefit. And we said, are they common, i.e. are they present in every sample? Are they shared, i.e. they're in a few of the samples but not all of them? Or are they unique, i.e. present in only one sample? And it doesn't show up very well, but what we found is that if you look at EGFR, which is that's a driver, it's something that stimulates proliferation, the cells to divide. If you look, these are the patients and these are the number of samples. So 5 out of 5 samples, 4 out of 4, 4 out of 4, they've all got EGFR amplification in every sample from one tumour. So that is likely to come from a common ancestor. Ditto, loss of P16. Every single sample. Others, like PDGF, is that up there? Yeah, PDGF somewhere? Yeah, there. Which a lot of people think is a key primary driver, wasn't present in these samples in every case. And we think it's a later event. But what it allowed us to do was it allowed us to look at the clones. And what I mean by that is one cell divides, gives off a population, like one family. And then another cell divides and that generates another family, and they're called clones. And each one of these little lines represents a clone from one sample, from one tumour. Big thick lines like that one mean that the clone is relatively common. But look at how many there are. There are hundreds, just in one small sample the size of a match head. Different patient, same issue. There is lots and lots and lots of clones. And what we think is that there's a founder which generates families, which in turn generate more families. And if we take samples from different parts of the tumour, they have different patterns of gene expression, but they're all present in one tumour. So some of the classifiers that are being proposed, some of the ways that we can distinguish one tumour from another, we're actually showing is not, probably not the case. So what's Charles Darwin got to do with it then? This is a picture of Charles Darwin's book, notebook, when he was working out the origin of species and how things evolve. And what we've done is we've started to think about how we might map that onto what's going on with all these hundreds and hundreds of clones. And what we think is happening is that all these clones are competing with each other. They're competing for resources, they're competing for oxygen, they're competing for food. And because they're fighting with each other, there's a lot of variability. We call it heterogeneity. So that in any one time, one clone is dominant. So if you think of a glioblastoma in one patient, one single patient, as the Earth in the time of the dinosaurs, you've got a dominant clone, that's the dinosaurs. Nobody messes with them. And that's the dominant clone. But if you treat this tumour and kill off that clone, what happens? Well, when the dinosaurs were destroyed, another clone arose. These mammals that no one had heard of. And one of the issues is that the clones that can evolve are often undetectable. They form less than 2% of all the population that's going on in a tumour. And they don't become manifest until we've changed the environment. And of course, we change the environment when we treat patients. If you give a drug or radiation into this system, it's going to kill off some cells, it's going to destroy some clones. And what's going to happen is new ones are going to emerge. And those are going to be selected to resist the drug, because that's the only way they can emerge. And that's why we think Darwin can help us understand why treatment fails. So where are we going in the future? I was asked to predict the future. Well, I think one of the things that's going to be important is that we're going to need combinations of therapies. We're going to need to be able to recruit patients properly from an integrated clinic, not just in Cambridge, but everywhere. And the NCRI are working to develop a national network of trial competence centres. We're going to need to administer drugs, look at imaging to see what's going on as you give those drugs. And then at surgery, we take samples. So we give the drug, then operate. We look at how that drug or treatment is affecting the tumour. And then we can then identify groups of patients for whom that drug or treatment will work particularly well and recruit them into clinical trials that have a higher probability of success. In other words, what we're trying to do is do what we do, but better. So I want to finish by looking at mortality from breast cancer, age 35 to 39. And this is data from the World Health Organization broken up into 10-year cohorts. And what's interesting is that from 1990 to 2010, in that 10-year cohort, the risk of death from breast cancer fell dramatically. There was no single massive biomedical advance. There was no sudden breakthrough in understanding what breast cancer was all about. There was improved delivery radiotherapy, improved chemotherapy, better surgery, and something we don't yet have for patients with brain cancer, which is screening. But it'll come. So if we can do that, then we can improve things in a practical sense. And I think that's what's going to happen in the near future. At that point, I'll stop. Thank you.
Colin, would you like to take questions?
Yeah, sure. Stunned into silence.
Got a couple of questions for you from Twitter. If you have that. Is there any evidence of brain tumours being exacerbated by air travel?
No.
One other one. What's the chances of survival if you've had PNET and been clear for 3 years?
Right, so a PNET in an adult?
All the information I have, I'm afraid.
Okay. PNETs in adults are rare, and they tend to be in young adults, and they tend to be quite aggressive. They're usually treated with a combination of chemotherapy and radiotherapy. In children, they tend to try and hold off with the radiotherapy, younger children. But the survival is not brilliant because they are quite aggressive tumours.
You mentioned earlier on that 59 patients would not have died if we were as good as the EU average and something like 225 if we were among the best. Do you know why? What is it that they're doing over there in the EU that we're not doing?
That's a very good question. I don't think we have a brilliant answer. If you go to a GP with a headache, you have a 1 in 3,000 chance that you've got a brain tumour. If you are, and so the GP is unlikely, I mean a GP will see maybe 1 or 2 brain tumours in their entire career. If you are French or German or Italian and you have a headache, you walk in off the street into a clinic, a neurology clinic, and you pay some money, and very often you'll claim that back from your insurance, but you pay some money and you'll get a scan. And you'll probably get a scan within 24 to 48 hours, maybe a week. That's completely different to what happens in the UK. That's one possibility. And that's not blaming the GPs, it's just a different system. Once you're in the system, the kind of organisation that we've got in Cambridge is by no means uncommon in many other countries in Europe, many other countries in North America and Canada. I think subspecialisation in neuro-oncology in the US began in 1994. The IOG guidance in the UK was published in 2006. Ongoing peer review shows that many centres are still struggling with even some of the basics in terms of getting the right infrastructure in place. Those are all probably contributory factors, and it's probably multifactorial. Not least, until fairly recently, we've not had a lot of data to understand the nature of the beast that we're dealing with. That's why we've worked with CRUK to get accurate figures as to incidence and so on. And it is a truism that what the NHS cannot measure, it does not treat. And therefore I think that's another factor.
You speak a lot about data. Are you hamstrung by the sharing of data? Can you share, like you were using, you referenced Eurocare and WHO, so are they quite...
As data sets, yeah, they're open data sets, yeah.
Okay.
Yeah. And we're establishing our own. The National Brain Tumour Registry is an open data set.
You do all your research and then that data is freely available?
It's not just the research, it's data on what happens to every patient. And what's happened is with the infrastructure's been established to pull the data rather than rely on other people to send the data in, which is fatal. So the ECRIC database people can just pull from every centre in the UK across lots of different platforms, the imaging, the clinical treatment, the survival, so on.
And what is that system called? Like, do you actually...
It's the National Brain Tumour Registry in the UK.
Colin, there's no doubt that Cambridge gives better service to some brain tumour patients for clinical treatment than other centres in the UK. So with your NCI hat on, how are we going to get the rest of the UK up to the Cambridge type level?
I think the key is building teams. I think it's about moving away from the attitude that we are all glioma surgeons here. It's moving away from... So when I was training, patient with a glioblastoma was reserved as a training case for the junior trainees because they were going to die, so it didn't matter if they caused a stroke and so on and so forth. And meningiomas, most of which are benign, non-malignant, were what the senior trainees got to play with. In Europe, most of the other European centres, it's the other way around. Trainees start with the simple meningiomas that are up here, and even very senior trainees are very closely monitored when they start operating on intrinsic brain tumours like glioblastomas and lower grade gliomas. So I think there's that element to it. And I think as surgery gets better, it's beginning to push the oncologists to up their game as well, so that we can deliver radiotherapy better, more accurately. And that there are techniques such as conformal radiotherapy planning, conformal delivery, intensity modulated radiotherapy, and the latest one that I don't really understand is called dose painting. But each one defines the target more and more accurately, and I think that's going to be key as well.
Building on that question, Colin, you say that you aim to get virtually all of your patients into a clinical trial of some sort, but that's very different in many other places that probably have few, if any, of their patients going into trials.
Mm-hmm.
What's the difference, and how can that be corrected?
So one of the things that we're looking at from the NCRI perspective is, can we identify centres that have a track record of recruiting patients? And by and large, they are the centres that recruit into EORTC trials. And there are about 15 of them in the UK. And I think what it boils down to is individual enthusiasm and motivation. And again, that gets back to the idea of having a team that works together. So for example, if you've got a bunch of surgeons who will work together, operate on a patient, see them afterwards, tell them the diagnosis, give them all the relevant information, they then go to see the oncologist, and they already know the diagnosis. Most people have gone and read it up overnight and looked at on the internet and scared themselves half to death. And they go to the oncologist, they know what's coming, they know a bit, and the oncologist can talk to them about the treatment they're going to get, about what the plans are, what the alternatives are, and about any trials that they're eligible for. If, on the other hand, you've got a bunch of surgeons who don't work together and they do an operation and they don't tell the patient, and the patient comes back to the clinic, the wound's okay, and off they go, and the oncologist tells them, then the patient arrives in the oncology clinic, they're told they have a nasty tumour, and they now, you hear the word cancer, and everything else blanks out. So for patients, we understand it's extremely frightening, and for the carers. So if you've got that fear as you're being told what's going on, your diagnosis, you're in no state to make any informed decision about joining a clinical trial or participating in research. So there's that kind of difference. It's about getting the teamwork in place so that when the oncologists come to it, they can talk about the trials that they want to put patients into. And the other thing that we're doing is we want to make it such that if there is a trial available, it opens automatically across the 15 centres. But there's an interesting paradox which we've found within the Anglian Cancer Network, which I'd like to share with you since you've asked the question. There are 3 centres that deliver chemotherapy and radiotherapy in the Anglian Cancer Network. One is Cambridge, one is Norwich, one is Ipswich. We have found, as we're trying to recruit patients into Gala 5, into Maltings, into several trials, is that patients who have to travel a long way would rather have standard treatment in their local centre than travel all the way down and join a trial, if that means they've got to keep doing that, because it's difficult. But that means you're taking a small population, the ones who are fit for radical treatment and clinical trials, and shrinking it further. We need to try and square that circle, and I don't have a good idea. If you come up with a good idea, anybody, I'd be very interested to know. Because at the end of the day, if patients are unwilling to travel, then we won't recruit them into clinical trials, and the pool from which we can draw will remain small.
brain tumours. And there's not many neurosurgeons in the UK that have that. Surely if we had more neurosurgeons who are basically at the cold face and are going to make sure that those samples go into research, we really would make a difference. So if there was more training, fellowship level, when the neurosurgeons are starting out, surely that would make a big difference.
We think so, and certainly some of our trainees coming through, you know, they gain more exposure to the kind of work that we're doing. It's more an exposure to a philosophy than practicalities. It's about the team-based philosophy and the philosophy that we need to to be rigorous in how we take samples. So as you know, Cambridge is one of the main training centres for training senior surgeons to use the PINC. And one of the interesting things was I was doing a case and my colleague Stephen Price was doing all the chin wagging in the lectures. And we bring them into the theatre to see what's actually happening. And I'd taken a piece of tumour out and I was trimming off all the non-fluorescent rubbish, because I know that if you take material and you want to do DNA and RNA and so on, and half of that material is crud, then you'll not get a good readout. So I trim off the crud. And I was trimming off the crud and I could feel the tension, because this lump was out of the head, it was sitting on a towel and I was trimming it away. And one of the surgeons finally tutted and said, well, what on earth are you doing, lad? And I had to explain why I was doing what I was doing. But until you've gone beyond that, then progress will be slow, but it will come. Sorry.
I came across what I think is the syndrome you're describing within my family, where the senior clinician was helping us, and at the end of the treatment, we exchanged letters. And I became conscious that this woman was an extremely good leader, and she ran a very good team, clinical team. And I'm wondering if you've understood that your leadership skills are special to you and they need to be taught.
That's an interesting.
They're not intuitive. Leadership skills are not intuitive.
I think you're right. I think it's. I mean, I've benefited from what's called the leadership development program that Adam Brooks runs, and I certainly benefited from that. I think you're right. I think one of the issues is getting buy-in from the relevant people, and that is going to take time, because first of all, as you're probably aware, you have the early uptakers, the ones who will pick up on something fairly quickly, adopt it, test it, evaluate it, and run with it. You'll have the majority who will wait until there's some evidence and then jump on the bandwagon, and then you'll still have that tail. No matter how much you do, they'll still resist change, and we just have to find ways of managing that.
Does there need to be some drive, sort of either sort of influence from government or legislative to kind of change the process? Because it almost feels like this is, you're relying on sort of an organic process and learning within the different medical centres within the UK, and unless you create a kind of a sea change paradigm shift, whatever you want to call it, it's just going to take years and years. And I'm just thinking back to your economic, there were some slides originally. I'm just wondering whether that, if you like, the commercial case for this and a better understanding of that would feed into that, the argument to...
I think I'll go back to what I was saying earlier on, which is that we are still in the process of defining the nature of the beast. We're still gathering that kind of data. I think as far as getting clinicians to do things differently, it's extremely difficult. If they are within the limits, that within the metrics that are there to measure performance and how they do, providing they're safe, and they're doing what they think is the best for the patient, it's very difficult to change practice. The only way we can do that is to present evidence over time, and there isn't really an alternative. You can certainly introduce protocols, which is one of the things that we're trying to do. And another thing that we're trying to do is by getting more centres involved in the clinical trials, which is very protocol driven, that also will inform their practice as they gain a greater understanding. A lot of centres who want to join GALA5 can't, because the surgeons and the clinicians just don't know how to get going. And they're often fobbed off, because if you've got a nurse who's funded from the cancer research, from the clinical local research network, who is primarily dealing with breast cancer, where she sees 2 or 3 patients for a trial every week, and she's then expected to take on brain cancer, where you might see 1 patient a month, she's not going to get involved. She's not going to be interested. She won't have time. And yet, at the same time, because we only see 1 a month, it's impossible to justify having a dedicated nurse, unless you get to a critical mass where your trial portfolio is big enough to justify that. And we're fortunate that we have that critical mass. But I mean, that clinic that I showed you, the pathway, the glioma clinic, took me 4.5 years to get that going.
Could I ask you on the science side, I mean, the fact that you can have a child that would develop, say, stage 4 GMB at 9 months, and then the adult would not show till 30, 35. How does, how does that evolve, that with the child it can move so quickly, and yet with the adult it can move so slowly?
I think it would be fair to say the bottom line is that we're dealing with different diseases. Just because they're called glioblastoma. Remember, one of the points that I was trying to make is that these diseases are named and classified based on what they look like down a microscope. And as we're starting to better understand the biology, we begin to realise that that's not necessary. What they look like might not necessarily be a good guide as to how they're going to behave. And I think that's particularly recently, there's been work coming out on paediatric glioblastomas, which shows that they are a different beast.
Colin, may I ask, specific to the therapies you're involved with in your trials, and I apologise if this is readily available information, you said that there were various patients who weren't applicable to your trials. With the fluorescence and with the wafers, are there lots of paediatric cases in your trials, or are they not appropriate for the wafers?
There's the lower age limit for GALA5 is 18. With regard to your question, there are some patients, when we first started particularly, who it looked like it was a glioblastoma at surgery. It was fluorescent. We sent a sample to the pathologists, and they came back and they said, this is not high grade. And then subsequently it turned out to be high grade. So we didn't put the wafers in, we treated them as a low grade. Equally, there have been some where they've come back and they've said, a patient has undergone surgery and the tissue has come back, it's not a glioblastoma, it's a grade 3, which also fluoresces. So they've had to drop out. That's the main reason that we've lost a few patients.
But there's no reason that paediatric cases can't be treated with wafers, essentially?
So there is a trial going on at the moment in Germany looking at using the pink drink in children. And the lowest age that we've done is a 17-year-old. But one of the problems is, outside of a clinical trial, it's impossible to get funding to use it.
How much will that usually cost them?
£900.
Is there any way that any research can be done or any data can be gathered about a patient pre-diagnosis? And I know that sounds very silly because obviously once you're diagnosed, that's when you start to take samples. But is there anything, has anybody actually tried to conduct questionnaires or look at previous tissue or blood samples that maybe have been taken over previous years and look to see if there's...
So the approach to that is called a genome-wide association study, or GWAS, and one of the trials that is ongoing is called the NBT study, and basically that is just a blood sample. And what they're doing is they're trying to gather, I think it's 3,000 or 5,000, I can't remember. And the idea is that study will then look at all the patterns of genetic changes and correlate them with phenotype. So that's one thing. That's looking at it from a genetic perspective. The other thing is, as I said, on that pathway, the before the referrer bit is an area that is extremely difficult to interrogate, because once a patient knows they've got a lump in their head, then when you start asking about symptoms that have led up to it, they have something called recall bias, which changes their perception. So it's by no means uncommon for a patient to say to me, oh, when I was 10, I banged my head. Is that why I've got a brain tumour? And it's probably not, you know. So what we need to do is we need to try and get to these patients when they are in a relatively naive state as far as disease awareness is concerned. And the only place we can do that is in general practice, in the primary care setting. And so that's what the Discern program is all about. It's about looking at a primary care setting. It's run by primary care, academic primary care physicians and epidemiologists. I was just involved because I was chivying them along.
When you pull the data and you're talking about all the different other centres that are involved, are there quite strict guidelines that other sort of centre of excellence have to follow in order that all the data is the same, where it's all collected, it's all...
Absolutely. So one of the key things is that everybody has to follow a trial protocol, right? And one of the key, just as an example, it's important that a patient has time to go away and think about joining a trial, all right? It's a process of consent. In essence, what that boils down to is they need to go and sleep on it, okay? So in order for that patient to be enrolled, they have to be seen, then they have to have time, then they have to be seen again, then they have to be operated on. A lot of centres, if a patient is referred in, they will keep that patient at the base hospital, wherever they were referred from, until there's a bed. They'll come in that night for surgery the next day, right? If you've got that scenario, for whatever reason, then you're never going to recruit anybody. Yeah.
Just talking about low grade, there's a few things that you kind of revealed which were new to me about the pink drink and the screening for genetic markers and things like that. One of the things with low grade, and certainly my experience, is that it's a watch and wait policy is balanced against an interventionist policy. Do you feel like watch and wait will happen less often now because of these new developments, as it were? I mean, I haven't even had a biopsy. I'm only diagnosed on MRI alone, and what you're saying makes me feel like I would want to go for biopsy earlier at this stage. So
what we know is that if you're an adult with a low-grade tumour, the vast majority of them are IDH mutated. Not all. And if you're IDH mutated, your tumour will behave differently.
So that's the kind of information I would want to know.
And so would we, because if you know that information, then that would potentially influence how you manage it. So if I had a young person with a wild type, low grade, we would keep a very close eye on them, 3-monthly MR. If we had a young patient with a mutated one, and we got a good resection, and we know the median survival is in excess of 8, 9, 10 years, then we might image them every 6 months. We'd do things differently. And that's only going to evolve. And that is basing it not on what it looks like down a microscope, but on a better understanding of the biology of the disease. And to do that, you need tissue.
And you need brain tissue. You can't get it from blood samples.
Not at this stage, not reliably, no.
You mentioned that there were age differences in some of your survival rates. Have you noticed any gender issues in terms of male to female rate? Or does it behave?
Other than that brain tumours tend to be more commoner in men. Other than that, no. The only other difference is that, particularly when you're doing the awakes, women seem to be much more stoical than men.
Yeah. And one last point. In terms of trying to diagnose earlier, has anyone done any looking at the environmental aspects? Because if you're a bigger centre with a bigger reach, whether there are some environmental issues that could lead to sort of predictive analysis of where illness may arise?
Again, it's very difficult because it's impossible to interrogate patients because they attribute, having had a head injury or been in a rugby accident or using their mobile phone or falling out of a tree or living up a mountainside nearer the sun's rays. And until we can do that when they're naive as to the fact they've got a lump, we will struggle to do that.
3 years ago there was quite a lot of discussion about interoperative MRI scanning.
Yep.
How does that compare with the work you're doing with the pink drink?
Okay.
And how long have you been doing the work with the pink drink?
Well, we've been doing the work with the pink drink since about 2008. We have one of the largest, about the third or fourth largest series in the world now. Is it better than intraoperative MR? Well, when I go to meetings and people talk about their intraoperative MR series and how wonderful it is about identifying a little bit extra tumour and taking it out and so forth, the thing that I always find interesting is they only ever get one intraoperative MR. They don't get a load of them. The pink drink gives you real-time feedback as you're going. It costs 900 quid. I think it's about 5 million to put an MRI scanner in. You need lots of fancy equipment. There's a delay, you need the patient, you need to prepare them. So at Queen's Square, I think their MR is downstairs in a basement somewhere. So they have to wheel the patient down, go down in a lift, and then bring it all back and start re-drape and re-operate. So I don't think for glioblastoma, for the high grades, I don't think it's superior to the pink drink. Low grades don't fluoresce, but they're a very small fraction. Their incidence is about 5 or 6 per 100,000. So it's very difficult to justify the cost on that alone.
So low grades being 1 or 2 rather than 3 or 4?
Yeah. What's interesting though is we've started giving the pink drink to low-grade patients to see if we can identify, and we choose the low-grade patients very carefully. In other words, are they at a risk that they might be changing into a high grade? And one of the things that we found is that occasionally what you find is you get a fluorescent focus inside the tumour, just a small area. And you would never know it was there under white light conditions. But one of the things we're interested in is if you take a sample of that specifically, would we be able to identify what's going on in that change?
Thank you.
Oh,
Sorry, I've got one more question for us. Part of the pink drink and wafer trial, how much would one patient cost as part of that trial? Because you said it's a 60-place trial, so from initial diagnosis through to end of life. And I know obviously the lifespans vary greatly, but as a main, how much would have been spent on that one person?
You mean in addition to what it would normally be spent? So basically what we're doing is we're taking a patient who would otherwise have surgery, radiotherapy and chemotherapy, and that would cost, on the basis of the EU data, around about £28,000, okay? The pink drink costs £900, and the wafers, packet of 8, is I think around £3,000 or £4,000. So it's about £5,000 or £6,000 extra. Sorry? Well, the surgical costs are pretty much the same, apart from at the end of surgery putting the wafers in.
My question was, I know you brought up the statistics for the Gala-5 trial, which was going quite fast. Could you just give us a quick sort of summary of the status of the Gala-5? I know you were thinking about finishing or hoping to finish.
So we need to recruit another 9 patients.
Okay. And how's it, in terms of the statistics so far, have you seen a notable improvement to the...
So the objective of the Gala-5 trial is to establish prospectively that it is safe to combine the pink drink with the wafers, so that we can then move forward and do a trial in which everybody gets the pink drink and then they're randomised to wafers or not.
Do you find it's extending?
We won't know that until the trial closes plus 2 years, and I'm not going to speculate.
Okay, thanks, Colin. That's very good. Just like to finish up on an optimistic note. We saw Colin's graph there of breast cancer and the incredible drop in increasing survival rates. So going forward for brain tumours, we have been starved of funding despite our valiant efforts, but things are changing. We've just issued a grant round for project grants to 10 institutions in the UK for 1.6 million. We're also doing another programme grant up £6 million later in the year. But in addition to that, after much lobbying, we've got Cancer Research UK to agree to nominate brain cancer as what their strategic cancer in their business plan from 2014 to 2019. And we'll be working with them on a number of additional clinical trials and also research projects. So I think things are really changing. We are going to have a massive increase in funding for brain tumours in the UK. We have seen the UK improve. When we started, there was hardly any research at all. I would now go as far as saying the UK is probably the second most active brain tumour research country in the world, thanks to people like Colin. The survival rates for the adults has been very stubborn, but in the last 5 years, the biological understanding of these tumours has been a massive increase, and that will eventually start being turned into increasing survival rates. So I'd like to thank Colin. You know, I wish there were a few more Collins in the UK Brain Tumour Society and everything, but I mean, we are getting there. We need more training, but I think we really will see things start to change in the next 5 to 10 years. But thanks very much for coming.